let's do neurons. Okay, so here's a bunch of nerve cells. The human brain consists 86 billion interconnected neurons. Okay, so essentially, you guys are all supercomputers. You guys are all more powerful than supercomputers, actually. So shown is an electromicrograph of neurons. Okay, anyway, so you are equipped with 86 billion neurons, interconnected neurons. Okay, so now you have sensory neurons. So your sensory neurons are bringing information in from stimuli. Okay, so from your sense organs, from your tongue, from your skin, from your ears, from your eyes, and stuff like that. Okay. They're bringing information to the processing center. So if there's not a lot of thought that needs to go in, then you just go to the spinal cord. So these are sensory neurons, then interneurons, and then motor neurons. They take out the processed information for you to react. Okay. So here's a nerve cell. This is the nucleus. This is the head of the cell. There's the axon. That's the long wire down which the electrical signal is transmitted. And these nerve endings connect to other neurons, so information can be passed from one neuron to another neuron. Okay. This is a myelin sheath. What that does is it conserves energy. Okay, so if you didn't have this myelin sheath, you'd need a lot more energy to transmit a nerve cell down the axon. But anyway, we won't worry about that. So that's sort of like an energy conserving thing. Okay. So this is showing you this dendrite, like I said, connects to another neuron, and here is another neuron connecting to this dendrite and this neuron. So these things connect to the head of the next neuron. All right. All right. So here is the axon. What we're showing here is, see the inside of that. We are showing you the axon. Okay. So here's the tube, and what is this? This is showing you is the potential. Okay. Now, it's so here's an axon. Okay. Now, because so here's the cell wall of the axon. Because of the properties of this thing, the inside is at minus 70 millivolts, and the outside is at 0 volts. Okay. So if you plot the voltage, so it says 100 millivolts, 0 volts, minus 70 millivolts. Oops. Yeah, okay. We'll say minus 70 millivolts. So the voltage inside is 70 millivolts along the length of the axon. So the resting potential is minus 70 millivolts. So that's what's shown here. Okay. And then once a nerve impulse is generated at this location, this is how the voltage changes. Okay. So the inside voltage has become positive for a small amount of time. Okay. There's the nerve impulse. So for a few milliseconds, for 2 or 3 milliseconds, the voltage inside has become positive. Okay. This is the nerve impulse, and that nerve impulse travels down this nerve impulse travels down the axon. Okay. So neurons transmit signals as voltage pulses called action potential. So this is a voltage pulse. Something changed here where the potential inside went from minus 70 millivolts for 2 or 3 milliseconds to plus 30 millivolts. Okay. And then that changes neighboring cell wall properties, and this pulse travels. Okay. So nerve signals, so this is a nerve impulse, and this travels at about 100 meters per second. Okay. So 10 times faster. The fastest human can travel is at 10 meters per second. So about 10 times faster. Okay. So this pulse travels about 100 meters per second. And the strength of the signal is conveyed by the number of pulses. So let's say you pinch yourself and about 10 pulses go per second. If you pinch yourself harder, then that would mean that 20 pulses will go per second. Okay. Anyway, I'm just making those numbers up. So the strength of the signal is conveyed by the number of pulses transmitted. All right, you guys can read all of that. Okay. Now, what we'll do is, so what I'll do is I'll show you how this 70 millivolts is generated. Okay. Okay. So the aqueous environment of the body is much like seawater. Its ionic solutes are mostly sodium and chlorine ions. Okay. So here's the axon. This is the inside, and that's the outside of the cell. Okay. And the outside of the cell is mostly sodium and chlorine. Okay. The inside is mostly potassium and large negative ions. Okay. So that's what's shown here. So here is the inside of the cell. There's the outside of the cell, mostly sodium and chlorine ions, and potassium and large negative ions. Okay. Inside the cell. These are ion channels and so on. All right. So inside the axon, the ions are mostly sodium and large negative organic ions. Okay. So there's a picture. The resting axon member is slightly permeable to sodium. Okay, so what we're saying here is the resting membrane is slightly permeable to sodium. It'll let in some sodium ions because the concentration of sodium ions is higher there. Sodium ions will, some of them will come in here. Okay. And this concentration of potassium ions is higher here, and some of these guys will leak out there via osmotic pressure. Okay. So the resting axon membrane is slightly permeable to sodium, very permeable to potassium, and always impermeable to the negative organic ions. Okay. So these guys cannot leak out. Okay. So the resting potential membrane lets in some of these guys go across, a lot of these guys go across. So when a lot of these guys go across, there's a negative charge here. And so that's how you end up with a minus 70 millivolt potential. Okay. So this leads to a minus 70 millivolts resting potential. Okay. Appropriate stimuli change membrane properties locally, leading to a sudden inflow of sodium ions lasting about 2 milliseconds. Okay. So here's how this nerve impulse is generated. Okay, so you change by pinching yourself or doing whatever, you change the properties of this membrane, and this becomes permeable to sodium ions. Okay, so there was a whole bunch of sodium ions, remember? See, there's a picture. Oops. There's a whole bunch of sodium ions. You do something to the membrane, and suddenly this becomes permeable to sodium ions, so the whole bunch of sodium ions come in. When a whole bunch of sodium ions come in, now the inside that was negative will become positive potential. Okay, and then the negative potential is restored by pumping out the sodium, and by more potassium going out, and then eventually the ions are pumped out again. But immediately afterwards, an increase in sodium ion flow out of the cell to restore the resting potential. Okay, so again, something changes. A stimuli will change the membrane properties, which will let sodium ion in, which generates this pulse. Okay, and then more sodium ions leak out to restore the equilibrium potential. So that's how this nerve impulse is generated. And then that nerve impulse travels down the axon. That nerve impulse travels down the axon. Once the nerve impulse is generated, it changes properties downstream, and the nerve impulse will travel downstream. Okay. So that's a nerve impulse. Okay. In an axon, charges move perpendicular to the nerve signal movement. Okay, so the charges are moving back and forth like that, and the nerve impulse is traveling in that direction. So charges are moving perpendicular to the direction of travel of nerve impulse. Unlike in a wire where the current flow and the charge flow are in the same direction. Okay. All right, so that was how nerve signals travel. And here's a little application, a pacemaker. Okay. So sometimes to make your heart beat properly, you have to send electrical signals at the right place. So when you send a little voltage here, this chamber will contract and so on. Okay. When you send a voltage pulse here, this chamber will contract. So your normal heart has the mechanism to do that automatically. Okay, so if you measure the voltage at various locations, so when a heart is normally working, this is this EKG signal you get. When it's not working properly, what you want to do is you have a pacemaker which will give electric shocks at the right time. Okay. So here's the switch for the pacemaker. When the switch is closed, when the switch is open, the capacitor charges, and then you close the switch and the capacitor discharges through your heart. Meaning, when the capacitor discharges through your heart, it's giving a shock at the right place. So let's say the first shock is there, and then the second shock will be there. So there might be two circuits like this. Okay. So it gives you a shock at the right instant. So there's with the switch open, the capacitor charges, the capacitor is charged. And once it's charged, then you close the switch and discharge it through the heart at the right time. Okay, so this is the current going through the heart. When the capacitor discharges, you have a spike in current, which makes the chamber contract at the right time. Okay. So again, so this is the kind of stuff you can do when you learn circuits and stuff like that. You can go device pacemakers and other such devices. All right, guys, so that ends today's lecture.